So we're going to talk a little bit today about, one thing I do need to add before I start. My background is aseptic processing and sterile filtration. And so I'm actually filling in for our subject matter expert, our virologist. So be gentle questions, and any questions I can't answer, I can take back. I have a handful of virologists that can definitely answer your questions.
What do you think about puffs?
I can tell you that one. Perfect. So we're just going to start out with, as soon as I figure out how to work the computer, which I'm getting to, just a quick overview of the agenda, a very high level ICH Q5 with the new revision that just came out at the beginning of this month. We'll kind of go over that, viral safety assurance, viral clearance, and then a very quick, very light selecting a virus, as I'm not the virologist. So history of ICH, basically the first one was published in 1997, and it was initiated based off of the fact that we were starting to see biological products where we were seeing viral contamination. And then in 1999, it was again revised, revision 2, just mainly for typographical errors. And then it was not looked at again until pretty much now. So in 2019, the ICH recognized that there were advancements in technology with manufacturing, with testing and basically analyzing and looking for viruses, as well as a lot of tribal knowledge that was gained throughout our manufacturing processes and our viral clearance techniques as we went throughout the years. And so essentially in March 1st of 2024, revision 2 was published. And it had quite a few, I don't want to say changes, but updates to it. It essentially allows for, highlights some key scientific principles, but it still allows for flexibility for the advancements of science as we move forward. It includes a risk-based approach, so it aligns nicely with all of our other documents that everything talks about risk-based and really looking at those risks associated with our productions. And then it addresses some new product types, which we'll get into in a little bit more detail, what's in scope, what's out of scope of the document, and then continuous manufacturing concepts, which I actually will not get into too much just because it's outside of the scope of what I do. So I don't want to talk about something that I'm already talking about something that I don't know very well, but something that I really don't know very well. So, and then it also discusses a platform approach for viral clearance evaluation as well, which it's interesting. It's using some of that tribal knowledge that we've gained over the years. So products within the ICH Q5 scope, you can see here, I won't go through all of them, but cytokines, monoclonal antibodies, which isn't anything new. However, genetically engineered viral vectors and viral vector derived products is fairly new. And you'll see that it's on the inclusions and the exclusions. So those that are amenable to viral clearance are included, and those that cannot withstand the viral clearance process are excluded. Some of the other items that are excluded from the scope of the document are inactivated vaccines, as well as some of our cell therapies. And it also addresses some new testing methodologies. Some of the, it really talks a lot about the next generation sequencing. It's one of the tests that it identifies the most throughout the document. And then of course it talks about the polymerase chain reaction. Those are the two main tests that it talks about. It does have a new section on molecular methods that I encourage you to look at because it does provide some additional detail on when you might use some of those methods. And then some of the other additional updates outside of the scope of what we're going to talk about. There's a large chapter that clarifies responsibilities for continuous manufacturing. There's also a discussion on the reuse of resins and prior knowledge of the impact of their use. So in essence, if you have prior tribal knowledge with regards to the resin maybe getting more effective with time, you can use that as a justification for maybe not doing as much validation testing for that end of life use of that resin. There's also a new section, an annex, detailing prior knowledge. This was a common theme throughout the document, really just using the knowledge that has been gained over the last 20 some odd years of biopharmaceutical manufacturing and viral clearance. There are some contingencies, such as the operation has to be very similar and well understood. The product intermediates also need to be comparable. And then it gives some different examples of some additional knowledge, such as knowing that what level of detergent is used for inactivation, or what pHs are commonly used and can be used for inactivation of the virus as well. And then it also allows for testing flexibility for well-characterized cell lines. It does use the CHO cell as an example, basically allowing for flexibility with regards to the number of virus or the log reduction that's acceptable for that particular cell line. Then we'll get into the nitty-gritty stuff. So we perform viral safety and viral clearance testing at our Bozeman facility, which is in the US. So Bozeman, Montana, we do quite a bit of biopharmaceutical testing. So why do we need viral clearance or a viral safety program? Essentially, a biopharmaceutical manufacturing process is extremely complex. There are a lot of steps involved with it. We're dealing with live cells, we're dealing with live biological systems, which are very variable and sensitive. So that's part of the reason. In addition, we're dealing with complex raw materials and starting materials. They're not always well defined or identified in, say, like the USP, where you might find a chemical, you can go right to the chemical page and look up exactly what you need to do the testing. That's not the case with some of these biopharmaceutical raw materials. Also, there's a risk of microbial or viral contamination in this particular process, because normally these have to be performed using aseptic processing. So that in itself is more of a risky process. You don't have a terminal sterilization point other than your filtration sterilization. And then there could be supply chain complexities. With cells, you're dealing with live biological systems. It takes a while to build up the amount of starting material or raw material you might need for your process. And so that adds complexity to your process and your processing parameters. And of course, because it is an aseptic process and you are dealing with those live biological systems, there is a lot of regulatory scrutiny. So in the ICH, the basic overall description of viral clearance or viral safety, you can really look at it in three pillars. One is prevention, so screening those raw materials up front. Detection, really assessing your worst case points within your process and screening for viruses at those points, so detection. And then removal, that final removal process of viral clearance, which is inactivating or actually physically removing the virus from the system. I'm going to say and you'll see why in a minute. So we'll start with the prevention aspect, which is when we look at the raw materials, these are the modes of contamination risk. So our raw materials, our starting materials, and our manufacturing environment. And so what we'll do is we'll look at ways that you might be able to mitigate some of the risks with these modes of contamination risks. So for instance, for raw materials, you might take a look at your raw materials and try to eliminate as much as possible. It's not always possible, any that originate from animals. In addition to that, if you're unable to use a raw material that does not originate from an animal, you can look at mitigation strategies such as heat, filtration, or irradiation to try to mitigate that risk a little bit. With your starting materials, you want to ensure that your cell baking facility is GMP compliant. We'll get into that a little more detail on the cell banking in just a minute. But there's also a possibility you might be able to use a virus-resistant cell strain. Not always, depending on what your virus risk might be, but that should be a consideration as well. And then also using well-characterized cells. So stuff that we already know, we already have a lot of information about it. We already know what the risks are associated with using it. That will help us mitigate some of the risks in the process as well. And then manufacturing equipment, as mentioned before, this is most likely an aseptic process. So using those single-use systems to reduce that contamination risk and or a closed system. So with regards to that cell line and the characterization and selecting your cell banks, you want to make sure your cell bank is using well-characterized cell lines and reagents in the process. You also want to look for a tiered banking system. So you want that working cell line as well as the master cell bank. So if one or the other get contaminated, there's another cell bank you can go to. And then you want to make sure that that facility is also doing robust testing of their cells and their cell lines. And so now we're to the detection portion. Overall, you want to look at the entire process in order to detect. So you look at the entire process and you look at your, from a risk-based scenario, you look at what steps might have the most risk associated with those. And those are the ones you want to target for screening and testing. So I have an example. This is not my process. I pulled this off the internet from the chemical engineer, but I love the process because it goes into detail and really labels all of the different steps within the process. So you can see I've identified where you might take a look at or where you should take a look at whether you have viruses within the system prior to be preventative, your cell line characterization, your raw materials, your lot release bulk harvest prior to purification, and then environmental monitoring throughout the process, and then your final product lot release. You will notice that there are two virus clearance or inactivation steps within the process as well. There's an inactivation step up above, and then there's a viral clearance step down below. And then some other tests you might want to consider as you move forward with it. Overall sterility assurance / viral safety assurance type of a program. You might want to look at some of your bioprocess residuals, extractables and leachables, which has been a key focus at this seminar. Environmental monitoring, facility process validation, so clean room disinfection validation, aseptic processing, as well as filter sterilization validation, which was also presented. And then final product QC lot release, characterization test, and final packaging test. So you see the whole list of tests and everything that we talked about with regards to viral clearance, but testing alone can't detect all of the viruses. And there's reason behind that. We just don't have a robust and adequate way to detect every single virus. And in addition to that, with virus testing, we're normally taking an aliquot of a large volume. So there's a volume factor involved with what we're assaying as well. And so you might have some low concentrations in which you may not be able to detect the virus. And then also direct testing may miss any target viruses. So if you're targeting a specific contaminant, you might miss some of the secondary viruses that are not specific to that tested kit, if that makes any sense. So how do we close this gap? There's a gap there, so how do we close it? And we close that gap with viral clearance studies. So initially, when we're designing a viral clearance study, we work with a customer and we perform a risk assessment on their process. And normally the customer comes in, they pretty much already know what their risks are, and they've already performed this assessment. But if they need help, we can help them do this as well. We're going to choose an appropriate spiking virus, and we'll talk a little bit more about how to do this, very high level. But essentially, it is taking a look at the process, what's included in the process, the ingredients in the process, the raw materials, and looking at any potential risk of virus contamination from those raw materials or starting materials. And then we're going to determine a clearance objective. And acceptance, meaning you don't just go into it blindly. You want to know what you want, what your process is eliminating. You want to have a target already established and not necessarily go into it blindly and think, okay, we're going to have 6 logs here and 7 logs here, and it's all additive at the end, but we end up not being able to meet that acceptance criteria. So we want to talk about that and have that established up front so that we can help you meet that need, whether that's adding another step or whether we need to change one of the steps. And then also you want to select your virus spiking positions and sample points. So with this regard, you want to be able to identify what your viral inactivation or your viral clearance steps are. And those are the steps we're really going to target and identify. And those are the areas where we're going to spike upstream and analyze downstream. So this is just a description of a couple different different activations or inactivations, ways that you can remove or inactivate virus. So removal and separation is normally performed by filtration, nanofiltration, or chromatography. My understanding from my reading is that the majority of the time chromatography is used. I believe it was like 70% of the time chromatography is used for this. Not sure if that's accurate. You guys can teach me. So, and then with inactivation, a lot of the times low or high pH is a common way to do the inactivation, as well as detergents and some high temperatures, low times. So not too much heat. So in order to have a successful viral clearance study, recommended not to just send us filters and the product and go, okay, do a viral clearance study on this. We want to make sure that there's appropriate pre-work that it's performed prior to that. So we should have a justification rationale for all the viruses that we're selecting to be performed for the viral clearance. And then in addition to that, really need to pay attention to the scale down process and make sure that the load capacities are accurate. I can't tell you how many filter studies I've had where we max out the filter and then it clogs and we have to start all over. So load capacities are important. The length of the process is important. And then, of course, the product stability. If we've got a process where our product is denaturing or falling out of solution, we have an issue. So we want to make sure we have this stuff determined prior to actually starting the viral clearance study. And then we also have a method suitability. We want to make sure that the product is not cytotoxic itself, and so it's not already causing issues with the analyses at the end on our cell line. In addition, we want to make sure that there's not any viral interference, meaning that as we're trying to assay for that virus, we can actually pinpoint and assay and quantitate the virus without any interfering compounds that might make it look like either it's lower or higher in the analyses. And then really take into consideration any shipping conditions, freezing, thawing, whether the product's staying stable. Those also come into play when it comes to designing these. This is my virologist would kill me. This is a really dumbed down version, but it makes sense to me because I'm a filter gal. So essentially this is a depiction of what a viral clearance study would look like, where you're adding the virus to one side at a known concentration that's fairly high, and then you have an output or a downstream sampling of the sample, and you're enumerating the virus downstream and determining a log reduction from upstream to downstream. At least 2 steps with 2 different mechanisms should be included. So meaning you shouldn't have an inactivation step and an inactivation step. You need to have an inactivation step and a removal step. So you should have one of each step at least. And then you want to choose more than one virus, and we'll get into that a little bit later as far as what the expectation is with how to choose a virus. And then normally we're looking for a high log reduction at each step. With the ICH, the new revision, it does allow some exceptions for some types of products. So that's you working with your regulatory body and having a thorough justification as to why we might not be able to meet the higher titer. And then Also, you want to use your worst case conditions with regards to your process. Somebody asked, what does worst case mean? And I am with you because I've seen some really, really worst case stuff come through that it is nearly impossible sometimes to test. So yeah, worst case conditions, process parameters should be demonstrated or used within reason so that, because we can always fail a test. I tell you, you can get a condition in which you can get it to failure. So yeah, within reason. So different assays that are used for virus detection. I'm just going to give you just a brief. I've already talked about qPCR as well as high throughput sequencing or next generational sequencing. There are pros and cons to both. So qPCR, it's well accepted for specific detection. So when you're looking for a specific virus, it's well accepted. It's very sensitive. It's rapid. However, it doesn't discriminate between live or dead cells. So it's problems counting both of them or looking at both of them. And it only detects what you're looking for. With high throughput sequencing, it's unbiased, so it's unselected. It can do direct identifications. It's well known to regulators, and it's very much pushed in the ICH document as well. The sensitivity can vary a little bit, and there's a little bit of a complex sample prep, so some additional prepping up front that has to be performed. With that, I also need to talk about the infectivity assays. You have to do not just an analytical method to assay to look for viral clones, you also have to do infectivity assays. They are considered the gold standard still. So that would be either a TCID 50 test or a plaque assay. There are other test methods that of course might be appropriate. These are the two most common. So some points that you want to consider for viral clearance, just to summarize what we've talked about, you should have more than one viral clearance step in the manufacturing process. At least 3 viruses with different characteristics should be used, which we actually haven't talked about that yet, but we're getting to that next. At least one step in the process should clear non-enveloped viruses, and you should distinguish between a removal step and an activation step as well. You should distribute viruses in different fractions, so identifying those key viral removal steps will help you do that as well. And then assay the process, assess the process steps individually if it's possible. So best case is to really single out those clearance steps and just remove that part of the process instead of having it go through an entire process. And then use orthogonal mechanisms to assure viral safety of the product, which were all the other methods we talked about with regards to viral safety. Now we get into the fun stuff. So selecting a virus for the clearance studies. So these are the 3 categories that the ICH specifies. A known relevant virus, which is one that you might identify in your reagents or your raw materials, or it's known to the cell line. Normally, they're, whether it's pathogenic or not, would make it relevant or non-relevant. If it's not pathogenic, it may not be relevant, and you might have a justification to not use that particular virus as being a relevant virus. And then they have a specific model virus, and this is something that's very close to the relevant virus. And we use this when the relevant virus is difficult to grow or we can't get it up to high titers. You might have to move to more of a specific model virus that simulates that. And then a non-specific model virus. And this is a virus that is completely different than, I shouldn't say completely different, but it's different than the viruses that you've identified within your solution. It helps give you a safeguard against those unknown viruses that might hopefully do not appear within your process or your system. And it has properties, different properties than the known virus has. So you're really looking at some variability in your virus spikes. This is just a beautiful picture of lots of different viruses. This is just to depict and show you the different sizes and shapes of virus. They're not all the same, and the sizes actually vary quite a bit. So some other factors to consider when you're selecting a virus, the step that you're trying to validate. You want to assure that you've got the right virus type for that. So for instance, if you're trying to validate a nanofiltration process, you don't want to use that big Ebola virus, right? You want to use this little guy over here, a parvovirus, because it doesn't make sense to take something really large and filter it out and go, yeah, my filter validation aspect works. So you want to consider the process that you're trying to validate as well. You want to look at whether the virus is enveloped or non-enveloped. Determine the virus particle size, which is what we just talked about. Also, the surface charge, you want to take a look at that, particularly if you're using a column. And then identify the genome, whether it's RNA, DNA, small molecule, large molecule. So this, basically what I did was I took different types of possible contaminants within certain processing conditions and proposed a model virus. And I did not propose the model virus, but I had help. So off to the left here is a possible contaminant that you might see within a CHO cell line, and off to the right would be a model virus you might be able to select for use in your viral clearance study. And this is the same with porcine material. If you have a raw material that maybe comes from porcine, you could potentially see any of these contaminants. And off to the right is a list of different model viruses that you might be able to use. And the same with human blood plasma products. And then this is just a general list. This is a common, I shouldn't say common, but typical list of viruses or virus spikes that we might see in our laboratory. So for biopharma products, we might see this list here. And then for blood product, we might see something similar to this. And it could be more or less different viruses. It kind of depends on the justification of the product. But this is a common grouping. And I'm done.